Thank you so much for coming tonight. Thank you to my partners with the National Park Service for giving us the support that we need in order to conduct this really important work. I used to live on St. Croix, and I worked for the Park Service for a couple years back. About a decade ago, we figured out this week, and so it's really nice to be able to continue this work and to come back to a place that I consider, a home away from home. So thank you so much for coming. I'm from Mote Marine Lab, and in the front we also have Dr. Abigail Clark. She's my postdoc, and Katie Eaton. She's my biologist. We're all in the Coral Health and Disease Program. Mote is a research institution, a complete nonprofit 501c3 organization in Florida. Our main campus is in Sarasota, where we have several hundred employees. But we often do work at this location here, which is our fortress in the Keys. It's on Summerlin Key. It's our International Center for Coral Reef Research and Restoration, where I'm the science director. So I get to oversee the 20 to 30 staff that we have there. And all of our staff are focused on corals, coral reefs, and bringing back the health of corals throughout the state of Florida and throughout the world. And so we're really lucky where we get to collaborate with organizations and places throughout the Caribbean, like here on St. Croix, whether it's research-based or restoration-based. And we look forward to being able to continue to work with the community to help preserve coral reefs, not just in the Florida Keys, but around the world. So again, thank you for coming. This is our field team wrapping up our 2 weeks here at Buck Island, and this is what you have in store for you tonight. So we're going to first talk about why it's important to care about coral reefs and why they're so valuable to us, how coral disease is a major threat to the U.S. Virgin Island reefs, knowing that this isn't an isolated location where this occurs. It's just embedded in the history of the ecology of this reef area. And then we're going to wrap it up by talking about what's happening out at Buck today and what we are trying to do about it to make sure that we have corals to last a lifetime. So why are coral reefs so important? Well, they are the most biologically diverse ecosystem in the oceans and probably in the world. They can rival rainforests on land. It's estimated that at least 25% of all marine life rely on coral reefs at one point or another in their life stage, and it's probably even higher than that. So biologically, they're incredibly important to our world, but they also provide ecosystem services. Those are things that they provide to us as humans. So one of those services they provide is they absorb wave energy. So as storms roll through, if you have healthy reefs offshore, all of the waves crash offshore and you have less impact from those waves coming in, you have less erosion, you have more protection of your property that you have right there along that shoreline. And so that provides a huge amount of protection as those waves are absorbed. They also provide a source of novel compounds. So whether you know this or not, there are medicines that we use today to fight things like arthritis, cancer, neurological disorders, and those chemicals that we are using in our medicines are harvested from organisms found in coral reefs. And so without that ecosystem there and without that diversity, we're actually going to be losing important novel sources of new medicines to fight additional human diseases. And they are a huge source of food for the world. At least 1 billion people rely on reefs for food and income. And that's especially true for a place like the U.S. Virgin Islands. So this may be, I'm sure it is a little bit dated. It's estimates from 2012. Coral reefs are a huge economic driver for any nation that lives right next door to it, like the USVI. So it's estimated that reef-related tourism brings in $96 million per year. $48 million are brought in through recreation. 35 million are through the amenities that people come into the islands to receive because of those reefs. $6 million in coastal protection, which I actually estimate is probably a lot more. $3 million in fisheries. And so that total estimate of how much that value per year of having a healthy reef out front of your neighborhood is $108 million. So the economic impact of having those reefs to the economy of the USVI is astronomical. And so preserving them is important for the biology, for the human services and economic services that those reefs provide to us, whether you see that every day or not. But unfortunately, reefs are changing. Zandy talked a bit about this during the last few minutes or so, and this is what a healthy kind of coral reef should look like throughout the Caribbean. It was dominated by elkhorn coral. Staghorn coral, which is a sister species of this, is a little bit skinnier, and that would have also dominated a lot of the reef area. And you can barely find reefs that look like this anymore, unfortunately. I mean, I can't think of one that I visited that still looks like this. Unfortunately, they look more like this. And these are iconic pictures of Carey's Fort Reef from the 70s to what it looks like now. And this is a place in the Florida Keys, which I call ground zero for much of what's impacting reefs around the world, but there are certainly locations like this in St. Croix that look very similar. Buck Island is, I think, handling things a little bit better, and so that's one of the best places I think that is doing today. But we are seeing this huge shift, right? You don't see a reef that's dominated by living corals anymore. You see a lot of rubble, you see a lot of bare, sandy substrate. It's dominated by algae. There's not too much fish because a lot of them have been harvested for food. And so the whole ecosystem has changed. And embedded in all of this is disease. So disease has been around in populations forever, right? It's a natural part of the ecosystem for every population. But over the last 50 years or so, especially in a place like the USVI, coral disease outbreaks have really changed the way that coral reefs looked. So this is a graph showing the historical presence of Acropora, so that's the elkhorn coral, during the Pleistocene and Holocene. So these are millions to tens of thousands of years ago. And they dominated the reef areas throughout much of the Caribbean. So this is a long-standing expectation within shallow reefs that you have the species dominating these areas. But what's happening is in the 80s and then now, we've lost those species. We've lost the dominant elkhorn. We've lost the staghorn. And the reason that we lost them is because of what Zandy mentioned earlier was white band disease. So this is an image that actually was taken by Philippe Mayor out here at Buck of white band disease. It's probably a bacterial disease that infects the base of the coral and moves up towards those branch tips. And it's that white line that you see of necrotic tissue that's killing the coral as it moves up from the base and out. And it often kills the entire coral. So it moves out until all the tissue is gone, and then you have a completely dead coral left. And that outbreak in the 80s and late 70s resulted in a population decline of over 90% throughout the entire range of the species. So that's from Florida all the way down to the southern Caribbean. This species was pretty much wiped out. And so we only have remnants of it left. And actually one of the best places where we still have some good populations is in certain parts of Buck Island. And this was actually first documented here at Buck Island and on St. Croix by the Gladfelters. And they're the ones that published this first peer-reviewed paper on the importance of coral diseases and how it's changing the ecology of critically important corals that are the foundation of reefs. So this is a publication from '77, aerial views of the northeastern part of St. Croix. It's hard to tell because it's black and white, but there's a dotted line here. And that dotted line is showing you that on the left are all corals that are bright white from dying from this disease. And the corals on the right are the healthy corals, but it's a pathogen that's moving across the reef. And that pathogen kept moving throughout all of the Caribbean, so that we have very few of these corals left. And then this is just a nice kind of image from that same paper showing how that disease progressed over time on the colony on the top left, resulting in complete mortality, whereas there's a colony right here that's surviving, right? So even as far back as the 70s, we see a lot of widespread issues, but survivors that maintain the population that we see today, which can be really important when you're thinking about restoration, right? because the corals that we have today have gone through a lot. And so they must be pretty strong. And so it's a good foundation for us to protect and to build off of to try and bring back these reefs. And now, unfortunately, there's not just one disease, white band, that's issues. There's tons of diseases. And this is just a handful of the most common ones that you would see if you popped into a reef in the Caribbean. So you'd be really hard pressed to go to a reef and see all healthy corals. They have issues, unfortunately. Some of them are, bleach. Some of them have discoloration. Some of them have tissue loss. But all of these diseases we see pretty frequently throughout places like Buck Island. So when you see a healthy coral, you want to see these beautiful colors, right? Whether they're oranges or reds or browns or greens. What you don't want to see is white. That means the skeleton of the coral has been exposed. And you don't want to see any discolored bruising or black areas that suggest that there's some type of an infection. But unfortunately, you do quite often. And then the history of disease outbreaks is a contemporary issue as well. So Jeff and I published this paper after a lot of hard work during the 2005 bleaching and then disease outbreak. So for those of you that were here in 2005, maybe remember how warm it was. It was one of the warmest years that we've had on record. The water temperatures were really warm, and we had a really big bleaching event similar to what you guys have just been through down here this past year. But what we saw was after that bleaching event occurred and the corals started to actually regain their coloration, they started to get infected with some type of a pathogen, and it resulted in over 60% coral loss of some of the major reef building species like the boulder and brain corals that are the foundation for the structure of those reef environments. And so disease isn't just in the past, it's in the present, and it continues to affect our reefs today. And one of the most unfortunate examples of a disease outbreak is one we're experiencing right now. So this is called the stony coral tissue loss disease. It started as what we all thought would be a small outbreak off the coast of Miami in 2014, where that red area is. The green area is just the entire Florida reef tract. So in 2014, it kind of popped up there. We all thought it would, kind of come and go as most of the other diseases occurred. This disease affects over 20 different coral species, so it's very nonspecific in the host that it targets. The elkhorn and staghorn are not part of the susceptible species, so that is one little shining star that we have, but it affects 5 of the corals that are listed under the Endangered Species Act. And these are many of the major reef builders, especially throughout the Florida reef tract. And this disease has continued to spread. So in 2005, it's moving north and south. 2006, it's gotten up to almost the northern part of the Florida reef tract and down into the Keys. 2007, it hits the uppermost part of the Florida reef tract and into the Middle Keys. 2008, it jumps over the 7 Mile Bridge, for those of you that know where it is, and now it's west of Key West. And really the only safe place are the western part of the Marquesas and the dry Tortugas in the Florida reef tract. So this has been an incredibly devastating disease, and it looks kind of like this. You can have a coral. This is a brain coral. That white is a little infected area that we saw in early March, and just a couple weeks later, the whole coral has almost died, and within just a few days, it was totally gone. And so this is a rapid tissue loss. So the tissue is becoming necrotic. It's basically melting off of the skeleton and just leaving a skeleton left on the reef. So if I'm a coral and I get a little infection right here, all the tissue of my body is just slowly sloughing off, and all that's left is my skeleton. That's basically what's happening to these corals. Most evidence suggests that it is a bacteria. Antibiotics is effective in treating it, which suggests it is a bacteria, but it's a waterborne pathogen. So that means that it moves very easily. It's super hard to contain. And then it also means that it can move pretty readily through human interactions as well, because people are moving water all around the Caribbean, right, through things like ballast. Here's another picture of some really large brain corals that you can see all of that tissue has just rapidly sloughed off. And you can kind of see that's a person next to it. So these are really large corals. And unfortunately, you know, it's not just impacting Florida now. It has jumped through many locations throughout the Caribbean. So it's extremely pervasive throughout the Yucatan in Mexico, Jamaica, off of the coast of the Dominican Republic, parts of the Bahamas, and then down island, right? So this is St. Thomas, and there's a couple other reports of it a bit east of where we are now. So it is jumping, not moving with the currents, it's moving into odd locations that could be related to human interactions and intervention within it. Okay, so the good news is that this disease, the stony coral tissue loss disease, has not been confirmed on St. Croix, so nobody has seen it here to date, but see the end of my talk because we'll talk about this a little bit more. So that's some good news, is that right now that's not something we have to worry about. But there is bad news. So, and this is Grumpy Cat. He's so grumpy, I know. So let's talk about the bad news that Grumpy Cat is telling us. And that is that some of those diseases that we've talked about are very, very prevalent throughout Buck Island Reef National Monument. So if you go east of Buck Island, I don't know, what is it, like 2 kilometers east or so, where we've been working, there's a lot of these huge mountainous star corals, right? And they are hundreds and hundreds of years old, and they have what's called yellow band. So yellow band is this kind of bleached area along the edge of the tissue that's really slow moving across the colony. It moves a matter of just a couple centimeters a year, so it is pervasive in that it remains, but it's very slow. So this gives us a chance to try some ways to get it better, right? And so we've identified a lot of these corals that are slowly dying from what appears to be yellow band. Black band is actually very common throughout the shallow regions around Buck Island. So there's reefs that are dominated by these brain corals, and they have what's called black band disease. So that's this black band that basically it's a consortium of bacteria that's dominated by a cyanobacteria. So that's the black part of it. That's cyanobacteria. It has these black pigments that's photosynthesizes. And underneath that cyanobacteria is a consortium of other kind of bacteria, and they all work together to create this really toxic environment for the corals. And the corals basically die and get eaten by the bacteria as it moves across the entire colony. What's unique about this is this is a very temperature-related disease, so it often only comes up in the summers. And I was super surprised at how prevalent it is right now in the middle of winter. Maybe because our winter right now is a little bit warmer than what has been in the past, and so this perhaps continues. And maybe more common with things like climate change and increasing ocean water temperatures. And then we also saw quite a bit of what we're calling acute tissue loss. So this is just a general description of tissue loss that we're seeing on a lot of the branch tips of the elkhorn coral on the South Fore Reef. And this could be from an assortment of things. It's just a very general visual description of some type of issue that could be an infectious pathogen or not, but one that we are interested in trying to study quite a bit more. So where we worked over this, the past couple weeks was really with these 3 diseases, the yellow band disease site that's kind of a bit far off the East Coast, and then in the really shallow, like 5-foot depth South Fore Reef, where there was a lot of this tissue loss and black band disease that we could work with. And then we also found additional sites that had a lot more of these same diseases, so this seemed to be geographically widespread a bit, and those sites are ones we're going to come visit the next time we're here, and I'll talk a little bit about more our plans for the future. So the objectives for our activities with this project are really to test novel methods to stop coral disease from progressing. So we have sick corals. A lot of these diseases are waterborne. You can't put a big, filter. You can't dump a bunch of bleach in the water. You can't dump a bunch of antibiotics in the water. So what can you do? Well, you can try to treat those sick corals like we do for humans in novel ways to try to get that coral healthy again. And ultimately, if we are treating those sick individuals within a reef, that should reduce that transmission rate among others and reduce the influence of that disease on a larger spatial scale. And as Andy mentioned, this is a 2-year project. So this year we're really focusing our efforts on the diseases that are affecting corals at Buck Island Reef National Monument. And then next year we'll be over in St. John. And there's a lot of different diseases and different issues affecting those corals. Even though they're kind of nearby, it's a different ecology. And so we'll have to, develop new methods and test new ideas out there. So the types of treatments that we've just started with, this is our first trip of 2 more that we're going to have throughout the year, are to start simple and then get into more complex applications. So the simpler you can make it and the lowest risk you can provide on side effects would be the greatest way to move forward. So if we can find a really simple mechanical way to stop coral disease from spreading, that's where we want to start. And so with yellow band disease, we are trying trenching, which is basically developing a fire break in between diseased tissue and healthy tissue. So again, if I'm the coral and my hand is infected, well, if I chop my arm off, can the rest of my body be saved through that, amputation, basically? And so when I started doing this work, there weren't these fancy underwater equipment that was available. And so we now have one where we can, take an angle grinder underwater and try these methods in a really easy way that's not incredibly time consuming. And so we're utilizing that type of equipment to do some of our fire breaks. And then we also use that underwater angle grinder to do amputation of those tissue loss that we were seeing on the branch tips of the Acropora. So a few years ago, I couldn't imagine trying to take a hammer and a chisel and knocking off just a lesion, because you'd break big branches off of an elkhorn coral and cause a lot of damage. And now you can use really precise, tools to remove the lesion and keep the rest of the coral intact. And then we're also really interested in using a 35% hydrogen peroxide solution on black band disease. So I kind of stole this idea from Aquarius. Aquarius used this as a way to treat cyanobacteria in fish tanks. So people that are really good at raising corals and raising fish in either aquaria, like in an official aquarium that is public, or like home aquarists, have used this technique for a long time. And so I heard about it and I thought, well, black band is a cyanobacterial disease. Maybe we should try putting this hydrogen peroxide on the black band and see if we can stop it from progressing. So this is a really common tool. It's an antimicrobial solution. It's much higher concentrations than something you would put on like a cut, but the same idea. It's just more concentrated. So if you put it on your skin, it would actually burn your skin at this concentration. Because it's so antimicrobial and used, but it's safe. The world uses it in the food industry quite a lot to wash fruit and vegetables. So they'll often be washed in a hydrogen peroxide solution that's of this grade. That's why it's called food grade, so that it can kill off harmful bacteria before we consume it. Yes?
How do you apply a solution like that underwater?
Yeah, we'll get to that. That's a great question. It's a challenge, I'll tell you that, but we're working on it. Okay, so we started out in 2015 trying this fire break method on yellow band, and we had a medium amount of success. So we're underwater, we're using hammer and chisel to just move along and create these fire breaks. And in doing so, we saw that we could reduce that progression by about 30%, which to me wasn't a high enough success rate, especially for the effort. I wanted to find something better. But we saw that the failure rate was often from these shallow, skinny trenches that we could make by hand because the skeletons were really dense. So often resulted in a regrowth and a reconnection of the diseased tissue to the healthy tissue before we could retreat. And so I thought maybe if we could be more aggressive in our trenching, using this new technology to create deeper, wider trenches and to isolate the healthy tissue a little bit better, it at least would be a good way to see, like, is this a method we could actually use to stop that coral from completely dying? And something that's super easy, there's no chemicals, there's no medicines that are needed to be involved. And so we tested that out this week. We took the angle grinder, and so this is kind of what it looks like underwater. So it's really loud, which is good that there's no audio on this, but basically you just kind of get to, if you're familiar with one, kind of place it along the right side of the lesion and move up and create a big trench between that light colored diseased tissue and then the apparently healthy tissue that you'd see kind of on the right side of the coral. So it's a little intimidating piece of equipment to use underwater at first, but we totally got the hang of it, and it was quite empowering to be able to zip along and hopefully make a good difference. So like kind of at the end, you can see I'm up here on it, and then this is what it looks like after we're done. So this white area is the trench. It's about a centimeter to 2 centimeters wide, maybe. It's about 5 centimeters from this leading edge. And then we just put nails in an area that's not treated and an area that's treated, so we can measure those progression rates when we come back and see if we can reduce the progression rate and prevent it from jumping over here. So we are creating we are creating an open wound, but all of the studies in the past suggest that that wound heals quite readily, and there's no secondary infections associated with this type of application. But
how deep is it? If it's about a centimeter wide, how deep is the...
It's about a centimeter deep, unless Katie has the angle grinder, and then it's about 3 centimeters deep. she got pretty aggressive, which is good.
Are you going to track the depth to see if that makes a difference?
Yeah, so there's a couple things we want to tease apart, depth, width, time of year. There's a lot of factors that could play a role of when this treatment occurs. And so that's kind of part of our long-term plan. If we see some promising results with this, we will need to refine how it's applied and the best practices associated with that type of application.
So it doesn't jump? On the surface,
Sorry, Ms. Swenson.
does that same thing happen in water?
Sometimes if we're working, like way down close to the sediments, but most of the time it wasn't creating big plumes as long as we were working just on the coral. And
It doesn't jump.
we haven't seen it jump per se, but we saw,
Somehow, so it
mhm,
moves through the water.
yeah,
1
yeah.
centimeter doesn't seem like a big gap for it to...
Yeah, and we'll see. So what we saw before is we would have this trench, right? And then this isolation would cause this tissue to die really rapidly. It'd die much quicker than a control lesion that wasn't treated. But then it would remain healthy over time as long as we retreated any lesions that formed. And then this was like the final one that we left kind of alone, which is still alive today. So I thought maybe it would be moving over, but there's a ton of corals out there that aren't sick. There's a handful of them that are, and some of them are the oldest ones. And so there's a lot that we still need to figure out with the disease dynamics and whether it is actually a contagious disease that's moving from one colony to another or something related to something else that's going on.
We live in Florida. I know in the Keys there was some program I heard several months back where they're planting disease-resistant coral.
That's us, yeah. Yeah. So we have, so specifically for the stony coral tissue loss disease that's throughout Florida, we have screened several of our genotypes, and we know some of them are highly resistant to that disease. And so we're using them in two ways. We're outplanting them very frequently, but we're also using them as parents for the next generation. So one thing you don't want to do is flood a population with the same individuals, right? Which is what you would be doing through clonality of corals. You're basically dumping a lot of clones out onto the reef. And that's a really important tool that we can use right now to get some corals out there. But the long-term goal is to create new individuals with high genetic diversity that have those disease-resistant traits to give them a boost in their ability to survive. So we're a part of that. I'm glad you've heard about it. We have a lot of work to do, but we're starting.
Yeah. Is there anything, or have you considered, like, possibly helping the coral to come to their own resistance of this bacterial infection? And then I also had a question as to maybe, sometimes we tend our homes for termites. Would there be something along the lines where you could isolate the entire piece of coral to help kill the infection and also maybe support their immune to that?
Yeah. So we actually have a couple projects going on now to, one, try to identify probiotics in the corals. So we're working with Kim Ritchie, who's a microbiologist and has worked with beneficial bacteria in corals quite often. And so in that sense, we would bag the coral and be able to treat it with the probiotic and boost their immune system, right? So that's one avenue that's in development, but quite a ways away from implementation. And then there has been other ways of, I think, of treating with antibiotics in that sense by bagging a coral. But what we believe is happening for the most part are isolated infections that aren't systemic. And so you can reduce the amount of antibiotics you use by topical therapy, which I'll talk about a little bit later. Did I get both of your?
I was just interested in having it
The beneficial
immune
boosting, yeah.
immune system boosting. And then I was thinking, would there be any, if you took portions of coral and did different treatments back in the laboratory to see if it was effective? Is that what you're doing with probiotics and or antibiotics? Which things might be the most effective?
Yeah, so we have done that a lot in Florida. We are partnering through this project with somebody from NOAA named Cheryl Woodley, and she's focused on doing the tank-based exposures of treatments and trying to come up with, there's a lot of different novel mechanisms that you can try in a tank, like bacteriophage, different types of antibiotics that have actually now transitioned into the field, and more pointed, riskier therapies that you could potentially use, but you need to start in the lab and then see what's effective and move out. So right now we are starting the simplest way, and if these mechanisms don't work while we're learning things in the laboratory, we can start using some of those that are more targeted and maybe more advanced in their therapy, but they also have risks associated with them.
I know initially you said that this isn't anything new that's been occurring for years. Several questions. Has there been an increase in the growth? And I know with the higher temperature, we can facilitate growth. Is that the reason? And if it is the reason, why have you, in one of your graphs, you showed a progression from north to south? I would expect it to be much more prevalent in the tropical area and progress north instead of the opposite direction.
Yeah, so climate change and temperatures can exacerbate disease, and we've definitely seen more disease outbreaks when water temperatures are high. One, because it could be affecting the virulence of the bacteria or pathogen that's out there. It also compromises the immune system of the coral because they get stressed and they're animals, and so like us, their immune system, is stressed and they can be more susceptible to infection. Now the stony coral tissue loss disease is a little bit of an outlier because it has no discernible relationship with temperature. It's all related to the epicenter, and it's moving north and south. It moved north faster than it moved south, and the Florida current pushes things right from south to north quite rapidly. And so because it's a waterborne pathogen, we think that's why it moved north faster, but it did move south and west. And it seems to be following these countercurrent eddies that show up at different times of the year. It's a kind of a weird one, and one that we have a lot of people working on because there is no correlation with high temperature. And so this one is a little bit of a new challenge for us to try to figure out.
Has anyone sequenced the genome of these diseases? I mean, are they all the same? Are they related? Are they different completely?
Yeah, so one of the challenges with trying to figure out the pathogen, right, is sequencing and molecular approaches are only correlative. So you can't figure cause and effect when you're just looking at what's there, because it could be there as a primary infection or it could be there as a secondary infection. So to do cause and effect, the golden standard is fulfilling what's called Koch's postulates. So you have to take a diseased organism, isolate the bacteria within a petri dish, take that bacteria, put it back onto a healthy individual and cause those same disease signs to occur, and then collect a sample and re-isolate the same bacteria, for example. And 99% of marine bacteria won't grow on a petri dish. And so we have done lots of molecular sequencing. We have hundreds and hundreds and hundreds of samples that we are processing for all different types of bugs, whether they're bacteria or other microbes that are there, what's going on within the coral, whether it could be related to like proteins and communication and chemo senses that are getting sent out. So we have a lot of people looking deeper in all of these diseases. But it's been a significant challenge to tease apart what's there, what's supposed to be there, what's there causing issues, and what's there as a secondary response to those issues. So it's really complicated. But, we're making strides. And the fact that sequencing is now relatively cheap, we can do more. So, okay. Let's see what else we're doing. Okay, so this is the black band treatments. And this is what kind of black band looks like up close. You can see that cyanobacteria black area that's adjacent to the tissue. And the white area on the bottom is that recently exposed skeleton. So I was not expecting this disease to be here, so we weren't totally prepared. We took a trip to the hardware store, got some syringes. Katie found some caps that go on the bottom of chairs with a little hole in it. Put some silicone on the inside and created a channel. We widened the hole so that the syringe could fit in, put the hydrogen peroxide in it, and basically held it against the disease and did squirts of it moving across the colony. So that was a very intensive, and this is kind of what it looks like after you treat it. So you get all of these oxygen bubbles that are forming, just like when you treat a wound, right? You can see that bubbling forming of the interaction between the hydrogen peroxide and the bacteria that are there. And so you get this bubbling that occurs, and a lot of the black band goes away. This definitely wasn't within 24 hours of us checking. It wasn't 100% that cyanobacteria was gone. But it definitely had a significant effect, and I don't know, we'll have to figure this out, with follow-up visits, how long it takes for that visual, sign of effectiveness to occur, because this is very much an exploration. So this is kind of what that coral looked like right after treatment. And then when we went back the next day, you could basically see that line, and there was teeny tiny bits of little blackness around it, but most of it had gone. And then with the acute tissue loss on the elkhorn corals, what that looks like is this. So this is kind of a zoom in of a branch tip. So this is a branch right here with the background of the base of the colony. And this white area is exposed. So all this tissue has recently died. There's a little bit of necrotic tissue here that's kind of fluffy looking when you see it. And so basically we would just kind of zip off that, that diseased area using an angle grinder. So this is just a little kind of image of, you get to get up close and personal, and you get to have really precise cuts with this angle grinder.
And Erinn, before you switch, go back to that very end of the video.
Uh-huh.
One of the things that's really important is you'll see there's a healthy colony right there in the top right, and you see the white edge. That's not diseased. For elkhorn coral, that is a healthy, living edge. So it gets a little confusing, is that, sometimes we'll get folks that come in from Buck Island, oh my God, there's white edges on everything. Well, in that case, and I'll let Erinn further describe, that's okay. But it's when it starts to go either band and go up or edge and go down. That's not growing healthy anymore.
Yeah. So these, on the ends of the palms, basically you'll see these white edges, and sometimes they can be several centimeters if the coral's growing really rapidly. So it can look alarming, but that is a good thing, because usually the coral's growing pretty quickly, which is usually what we want. So here you can just see that the branches lopped off. That diseased tissue is removed from the reef. We bring it back to land. We bleach it so that all of the tissue and the bacteria are killed, and all the microbes are killed. And then that piece of skeleton just stays on land in case there's any, just as a precaution. Okay, so that is what we've done. Next time, what we're going to do is we'll come back and we'll revisit all of these corals. We'll be able to look at our success and failure rate based on these really initial testings of some of these pretty mechanical applications to see how effective they are. And we have controls for each of those. So we treat a certain number of corals, and then others we leave them alone, and we'll see how those two compare, whether there's continued tissue loss on the controls and a cessation of tissue loss on the treated corals, or a very comparable situation. We'll be able to look at that the next time we come in. I also am interested in trying to test the antimicrobial properties of the hydrogen peroxide on acute tissue loss and yellow band disease. If it is an effective antimicrobial compound, I think it would be a very low risk application that we could use to try to treat these diseases if they're microbial pathogens in nature. And then we can also test a lot of the new developments in these topical therapies. So with the explosion of the stony coral tissue loss disease from 2014 to today, that allowed people to start thinking more progressively about what we're okay doing to try to get corals healthy. So about 5 years ago, antibiotics would be off the table. But we're at a place where if we don't start doing something, in assessing and balancing the risks associated with doing something versus doing nothing, we're probably not going to have any corals to do anything with in the not too distant future. Oh, that was the first time. Nice. So a lot of the laboratory tests are showing that the tissue loss diseases they stop when you get treated with antibiotics, when the coral's treated with antibiotics. And you can mix that antibiotic with a delivery vehicle like a paste and just spread it on that lesion, and it can be very, very effective. So you can be targeted with a low dose of antibiotics that is extremely negligible compared with all the antibiotics we put in the water through the flushing of our systems into the sewage, into the waterways that don't get treated in any of the municipalities, right? So every time we take antibiotics, they actually flow out quite heavily into our nearshore oceans and waterways. And so we're putting in a ton more antibiotics than anything we would be doing by treating coral diseases. And so that was a study that actually happened in Florida to show the risk was extremely low for what we're proposing to do to try to treat these corals. Chlorine is another option that has been effective on some diseases in Hawaii. So that's another, like an antiseptic that is not an antibiotic. So the risk, of course, with antibiotics is producing resistant bacteria. So chlorine is an antiseptic, and so it wouldn't be creating or targeting these resistant genes that could be proliferating after antibiotic treatment. And then there's other antimicrobials. So there's exploration right now with lab experiments looking at essential oils, because some of those are effective as an antimicrobial. And so looking at natural products and seeing, could we try some of these and see how effective they are, so we don't have to use antibiotics all the time, and start exploring our options, to reduce the risks and increase effectiveness. And then I also need to develop a better hydrogen peroxide delivery mechanism. So something that's stable on the coral, that's time efficient, and that creates a concentrated dose for a prolonged period of time. So I'll be working with somebody in Sarasota that's already contracted to do this. We just weren't ready with the prototype today for this trip because I didn't think it would be such a big deal. And then for those of you that may be familiar with whitening gel on teeth, like in a lot of my searches, 35% hydrogen peroxide is sometimes the concentration that's used in gel for whitening teeth in dentist's office. And so it obviously can be integrated into a gel, which would be a lot easier to use than a liquid that's in a liquid environment. And so I'm going to be exploring the integration of that into a gel that could be used, really easily underwater compared to other options that we may have to use if not. Okay, but remember I said, listen, there's more at the end about what's going on with stony coral tissue loss disease. There's something going on at the underwater trail. So the first day we were here, as Andy said, I want you to go out to the underwater trail and I just want you to look around, because if stony coral tissue loss disease is going to come here, that's probably going to be where it shows up, because it seems to be hopping with people, right? And that's like a really high visited location. And so that's probably pretty true. And I didn't see anything that was like, freak out. But there were some corals of concern. So this is one coral that I saw the first day we were out there. It's one of the more susceptible species to tissue loss. It does have tissue loss. It could be something completely unrelated, or it could be the first signs of something that we need to be concerned about. And then this is it just a couple days ago. So you can kind of see some real clear markers here that are, the tissue has surpassed quite a bit. And so it's moving extremely rapidly. This coral, if it progresses, will be dead in just a matter of, a couple weeks if it continues at this rate. So, that's something that we really need to keep an eye on. And then right next door was another coral of the same species that had some more tissue loss. So less dramatic, but here's a little lesion that's got bigger, probably 5 centimeters bigger in just a couple days. So that was, they're right next to each other, so that is something to be aware of. And then these, and that's a close-up of it, just to give you a little bit better view. And then the SID. So this is, I don't know the common name of it. Do you know the common name? You know the common? Nobody, we don't know common names. That's terrible. So it's a big boulder coral. It looks like a golf ball, little pinholes in it. When we showed up, it had this bullseye pattern, which is really weird. So the bleached kind of area is showing up in these rings. And I only saw one coral look like that, but we have seen corals look like this in the Key West region right as stony coral tissue loss disease was hitting. So it's this weird phenomena that we call the bullseye pattern, and I saw that right next to these other two. So, you know, what do we recommend? I think at this point, it's good to take precautionary measures. This could be nothing. It could just be, you know, some sick corals caused by something that happens, you know, every day, unfortunately. But I do recommend that the Park Service does, and I think they did, treat with topical therapy. So getting out there and using that amoxicillin mixed with the delivery vehicle that treats that lesion. And that has been shown to be extremely, extremely effective in Florida. And so that has been done today. And to watch this area closely. And this could be, you know, as I said, you know, a false alarm. But this is a good time to get people talking about what to do and to get a plan of action in place and be ready to mobilize. Because what we have seen in Florida is that when it hits fast. And one of the benefits of us having gone through what we've gone through is the lessons that we have learned. And it took Florida years to mobilize, years. And we've lost what little coral we have left, basically. We're at a 2% coral cover now. So this is a lesson to learn. You know, you guys could be ready to be mobilized, and it's really hard to contain a waterborne pathogen, and it's going to be hard to make a big difference on the entire Virgin Islands. But now's the time to get a plan together and figure out what can you do, what do you need to do it, how do you make that happen, and get a plan of action together so that you fight as hard as possible to preserve what there is left, because these are the corals that we need to keep alive in order to, you know, recover what we've lost. I don't, you know, I want my children to be able to experience the amazing world of coral reefs. Like, my grandmother used to ask me, like, where's your favorite place to go? And I would say underwater, because that was where the magic happens, right? And so I don't want my children to lose that. And so we have to think of these novel ways. We have to be ready, and we have to save what's left, because that's a foundation for rebuilding. So I want to end the talk by recognizing some of the youth that we have in our audience. Would you guys be willing to just stand up and wave at everybody? So luckily, you know, we come down here, we get to do a variety of things. We get to try to save corals, but at the same time, we get to connect with the youth of the island through the junior scientists in the SEED program. And that's what these guys that are here are a few of them that are becoming the voice of the community of the youth. So they are getting scuba certified. We all got to dive with many of them on Saturday out at the pier. That's where we were. And they're amazing underwater, and they are, you know, becoming a spokesperson for coral reefs conservation, and, you know, making sure that the community is aware of the gem that's surrounding this island, and, you know, will be a huge source of leadership as they continue to grow. And they impress me immensely, and I'm really lucky that when I get to come here that I get to work with you guys. And thank you so much for coming tonight. It's good to see you. You're welcome. So with that, I want to thank the Park Service especially, Nate and Kristen for being the muscles and the maneuvers behind all of our activities this week, and Abigail and Katie, who are the ones that are, we're working together underwater to hopefully make a change. And of course, we couldn't do it without all of these people that are really helping to get us the support that we need, whether it's financially or logistics, to make this project happen. And I hope that as we continue to come down, we get to report good news of discoveries that we've made to try to save the corals that are out there. So again, thank you all for coming, and I'm happy to answer any questions.